All right, let's dive into something truly fascinating today. The place where artificial intelligence and cancer therapy are coming together. We're going to explore a brand new way to fight cancers that, up until now, have been almost impossible to touch.
So think about this for a second. This one question has been a major roadblock for cancer doctors for years. You see, some of the worst cancers are caused by faulty proteins, but these bad actors are hiding deep inside the cancer cells, basically untouchable. So what do you do?
To really get a handle on the solution, we first have to understand the problem. Why are these cancers so incredibly hard to treat? Well, it all boils down to a kind of fortress at the molecular level.
Okay, so here's the fundamental challenge. Most of our best antibody drugs are, well, they're like big, bulky security guards. They're fantastic at patrolling the outside of a cell, but they're just way too big to get through the door, the cell membrane. And that means any cancer-causing troublemaker working on the inside is completely safe from these drugs.
And inside that fortress, you find the real masterminds, proteins with names like KRAS and P53. Think of them as the cancer's internal control panel. When they go haywire, they're essentially flipping a master switch that tells the cell to just grow and divide like crazy. And for diseases like pancreatic cancer, more than 90% of them are driven by these KRAS mutations, all happening inside that protected space.
This whole problem is so significant that it actually has a name. It's called an undruggable target. This just means it's a protein that causes disease, but our normal drugs can't get a grip on it. Either it's hidden away or it just doesn't have a good spot for a drug to latch onto. For a long, long time, this has been a huge dead end.
But what if this fortress has a secret weakness? A little telltale sign that gives away exactly what's going on inside. Well, it turns out it absolutely does. Nature has given us a really fascinating loophole.
It might sound a little strange, but every single cell in our bodies, and that includes cancer cells, has this kind of built-in quality control system. It's constantly taking little pieces of whatever is inside and showing them on the outside for the immune system to check out. So the cancer cell can't help but advertise its own corruption right on its front door.
So here's how that works, in 4 pretty neat steps. First, those bad cancer proteins inside get chopped up into tiny little pieces called peptides. Second, those pieces get ferried up to the cell's surface. Third, they get placed into this special little display stand called an MHC molecule. And finally, this whole package, the peptide and its little stand, sits on the cell surface like a tiny flag basically screaming, "Hey, immune system, look what's going on in here."
So that raises a really powerful question, right? If the cancer cell is raising these tiny, super-specific flags, can we design a weapon that recognizes only that flag? And that is exactly where this revolutionary AI approach comes in.
So, let's meet the hero of our story, the TCR mimic antibody. You see, our own immune system has T cells that use a natural sensor to spot these flags. This new kind of antibody is engineered to mimic that ability, but with way more precision. It's basically like a custom-made master key designed for that one specific lock on the cancer cell.
And this is a complete paradigm shift. The old way of finding drugs was this slow, costly, trial-and-error game of just screening millions of things you already had. AI flips the entire script. Instead of looking for a key, it designs the perfect key from scratch. It uses these predictive models to build a brand new molecule, perfectly tailored for the job. It's a massive, massive leap forward.
So this all sounds amazing in theory, but how do you actually make it happen? Well, this whole project lays out a really clear 4-year mission to build the engine that can do just that.
This chart here lays out the whole 4-year roadmap. I know, it looks a little detailed, but the key thing to see is how all these different efforts are happening at the same time. They're building the AI, gathering the biological data, and perfecting the lab techniques all at once. The point isn't just to find one drug. It's to build a repeatable, intelligent platform that can create these master keys for all sorts of undruggable targets.
The mission really boils down to 3 elegant steps. First, you build the library. That means collecting tons and tons of data on which flags are actually showing up on cancer cells. Second, you design the key. This is the AI's job, to invent the perfect antibody for that flag. And third, you have to test the lock. The scientists take the AI's blueprint, build it in the real world, and make sure it actually works. And what's so crucial here is that this isn't starting from scratch. These results you see here show that the team already has the expertise. For example, those charts on the left, that's them already showing they can tell the difference between the good signals and the bad signals from those little flags.
This kind of foundational lab work is what proves this ambitious plan is more than just a theory. It's a viable strategy.
But maybe the most brilliant part of this entire project isn't just that they're using AI. It's that they're creating an AI that gets smarter with every single experiment.
What makes this so incredibly powerful isn't just one prediction. It's the creation of a self-improving system. It's a closed loop where the AI learns from its successes and its failures, getting better and faster and smarter over time.
So here's how that cycle works. The AI makes a prediction. It designs a key. The lab goes and tests it. Then all that new real-world data gets fed right back into the AI model. And that means every experiment, even the ones that don't work, is a super valuable lesson that makes the AI better for the next round. Its next predictions are going to be even more accurate. So after 4 years of running this intelligent loop, of building the data library and training the AI, what's the actual tangible goal? It all comes down to one thing: producing one powerful preclinical candidate drug that's ready to move to the next stage.
And this is key. This is not just an academic project. The whole point is to turn this amazing science into something with real therapeutic potential. To take this what-if idea and turn it into an actual drug candidate that could one day make a real difference for people with these incredibly tough cancers.
And Matches leaves us with this final, really powerful question. If this platform works, it won't just create one drug. It'll create a blueprint, an engine that can learn and adapt and design custom therapies. So you have to wonder, could this be the key that finally unlocks a future of truly personalized medicine? One tailored not just to a type of cancer, but to the unique molecular fingerprint of your own tumor.